analyses, aligning sequencing reads, colon variation, gene annotation, variation interpretation, very major examples of how we use reference sequence. In terms of types of sequence, we primarily think about DNA, but there is no reason to consider, no reason not to consider mRNA, cDNA, CDS, protein sequences or peptide sequences in this exact same idea of reference sequence. So to concentrate a little bit on reference sequence in genomic analysis, this is a download of a GA4GH germline caller from the GA4GH website. This forms a best practice. And I just wanted to highlight one area up here in the raw unmapped reads area under position 1. The second step of this pipeline is map to reference. So you can see the usage of the word reference appearing, but really reference sequences are so fundamental to this process that without it, the rest of this germline caller would not exist. Effectively, what we have done within bioinformatics and genomics in general is to now declare variation as a difference from that baseline reference. So we technically don't have variation unless we have a reference. Now, one of the other interesting attributes of reference sequences is that we can use them to compress sequencing reads. This is a screenshot from the Ensemble project website, which is a project I'm heavily involved with in my day job. It's a alignment of the Illumina body map to RNA-seq project to a region of GRCH38, which some of you may know as HG38. And what I wanted to highlight is this little area here. Everywhere on this screen, which is gray, is an alignment which is identical to the reference sequence. Everything that is highlighted in a different color, such as red, is a difference from that reference sequence. And you can see in some of these columns here, nearest to where the left-hand side of this blue, light blue box, there is a read base pair that says G and one that says N. If you look very much at the top of the screen there, you can see that both of these positions on the reference sequence are both T's. What this allows us to do is that we can actually create formats, and Global Alliance has already created a standard format called CRAM, which fundamentally it uses this, this attribute of sequencing reads to reduce the amount of storage required to store a read. Basically, you just need to know the differences between a read. You don't need, and its reference, you don't need to know all the base pairs of the read. Now, if you're going to do that, there are some major consequences of using the wrong reference. Here's a brief toy example. All these 3 sequences are all chromosomes. They are from GRCH38 or HG38, GRCH37 or HG19, and GRCM38, and I think that's MM10 in UCSC. We have a example 5 base pair read at the top left, which has a run of 3 reference base pairs, a base, and then a 5th reference base pair. And I just want to highlight the exact same regions here in this left-hand column, and you can see in the first example from GRCH38 that actually that example compressed read reads as AAGAT, which is identical to the reference sequence. That suggests that this is the wrong genome to be using for this reference sequence. The second one, which is probably the most likely because we said that this was a human read, we assume it's a human read. We can read through the reference sequence there and we can see the read is actually TAAAT, and there is a difference from the reference genome sequence in that 4th position, so this looks absolutely fine. Now the thing is though that we could actually introduce the mouse genome here at the very bottom for the exact same region, and suddenly the read changes from being TAAAT to GAGAG. Now which one of these is right? Obviously, we can exclude the first one because it's identical to the reference sequence. Something would have gone wrong in our compression language if that was correct. But these other two sequences both are technically valid. The only thing we could say is, well, we know this is meant to be human read, so we should actually use the human reference. So keeping things such as a chromosome name and location is insufficient for reconstructing this read. We need to know something more about that particular baseline reference sequence. We could use assembly, but again, assembly names can be problematic. So naming is always a problem. This is a slightly modified quote, but from Phil Carlton. But I do like it, that there are two hard things in computer science, and I would definitely say genomics as well. We have off by one errors, we have cache invalidation, we have naming things. Naming things is just difficult. So one way of solving the previous problem that I mentioned about using chromosome names is, well, we should just use a common name, which is fine, but already within the genomic space, we've seen projects such as Ensemble use a naming convention of 1 to refer to chromosome 1. And UCSC and NCBI have, I say recently, in the past few years, have switched over to a nomenclature using CHR1 to define chromosome 1. So there's already a difference here in the wild of what's actually being used. We could use a pre-existing universal naming system or an identification accessioning system. INSDC has such a thing as this. All genomes should be submitted into INSDC should they be available for public consumption, and INSDC will mint an accession and version that accession as well. One issue with this, though, is that there's no way to actually verify the identity of the sequence. So again, we're back to that same problem. If getting the wrong read, the wrong underlying sequence is so fundamentally wrong and bad for reconstructing the sequence, we need to get the right sequence. And in a way, it's very much similar to the 419 scams that were flying around. I claim I'm this particular person, but how do I verify that that person is real? So given that identifier there, I have no way of verifying the identity of the sequence that was used. So the people involved in CRAM, at least, decided to suggested a new identifier space that can actually be verified. One way of doing this is using a nice little algorithm called a checksum. A checksum, for those who are not aware, it's a very small algorithm that can take in a stream of data and then generate another small payload of data that can be used to verify data integrity, by which if you apply the same stream of data through the same checksum algorithm, you will come out with the same answer at the other end. Now, that does not allow you to verify data authenticity. That you could have someone say, oh, of course, here is the checksum, but you would have to then recompute the checksum in order to actually verify the data integrity. But you can verify data integrity through it because it's fundamentally part of the way that checksums work. So down at the bottom is a rough idea about how we generated this new identifier system. We have reference sequence. We pass it through a normalization algorithm, which is pretty much retain only uppercase A to Z characters and remove any other character from the sequence that doesn't conform to that. We pass it through a checksum algorithm. We have chosen MD5. There are very good historical reasons for why we chose MD5, which I'll go into in a minute. And then this gives a nice condensed hex representation of that checksum at the very end. So Refget, in a nutshell, is a way to retrieve reference sequence by a checksum derived by the contents of the sequence. It's very meta. I do apologize, but this will begin to make more sense in a minute. But I'm going to take a brief break from that. I'm going to now dive into where Refget's place within Global Alliance is, and a little bit on Global Alliance. So Global Alliance is formed of a number of driver projects, which intersect with a number of foundational and technical work streams. Real-world driver projects are meant to be genomic data initiatives that are basically there to help guide and to pilot our tools. They are there to tell us what is needed by people who are attempting to bring genomics into healthcare systems. The foundational and the technical work streams receive that input, and they attempt to then generate specifications, APIs, implementations that will help to actually serve those standards. On the right-hand side, we also have partner engagement, which basically means that we can continue to engage with the international community, with national health initiatives, and also with patient advocacy groups as well. You can see as well the foundational work streams breaks down into some other very key areas. One of these is regulatory and ethics, and also data security. So obviously we are talking about personally identifiable human data in a number of these cases. We are talking about very sensitive issues as well when we come to healthcare. So these have to be at the forefront of our thoughts. So the foundational work streams help support the technical work streams to ensure that we are not only conforming to regulatory issues in countries, we are also ethical in how we are behaving and how our APIs will work, but also that we are secure in the way that we are developing our APIs and specifications. Of the current driver projects, we have a fairly broad remit of driver projects available, which span Europe, they span the US, and some of them also span Australia as well. Currently, I believe there is still an open call for new driver projects. If you are a representative of a group who would like to be a driver project, I would sincerely ask you to contact the Secretariat and also consult the Global Alliance webpage for more information on how to go through that particular process. At the moment, you can see how these different, this is the Global Alliance 2018 Strategic Roadmap. Down the left-hand side, you can see a number of deliverables. There were 28, not all these were delivered over the past year, but quite a large number of these were delivered. And you can also see that each of these is colored in the various different work streams. And if you look down the left-hand side, you can see number 21 is Reference Sequence Retrieval API, which is this particular API, Refget. This was written before we renamed the API into Refget. And you can see that this is actually formed of two work streams. Large-scale genomics is the primary work stream for Refget, and genomic knowledge standards, which is my work stream, is heavily involved in this, obviously through my own participation, but also through the major focuses of both these work streams. Large-scale genomics is attempting to look at issues concerning data storage, data dissemination for primary analysis of genomic data. So they are the guys that are creating things such as the CRAM format and the maintainers of the BAM and SAM specifications that are available in the wild. GKS, Genomic Knowledge Standards, is very much interested in variation. And obviously, as I said earlier, we can't have variation unless we have a reference. So we both understand that this is essential to our future work. In terms of where we sit within the overall structure of how foundational Refget is, you can see it's at the very bottom here of discovering data. It is in the area of genomic and phenotypic data transfer. It's also part of the discovery data component as well. So this is very much about data that we're providing. Refget is not about running software, it's not about user access. It is not about how we represent genomic and phenotypic and clinical data. And very much at its core, Refget is also reinforcing ideas of FAIR as well. So Refget, reference sequence retrieval, is about the findability of that data. Because we will have a reference sequence with the checksum, you will be able to find that data. It is also actually part, some of the future developments as well that we'll have will target areas of accessibility, and also will target areas of interoperability as well between different services. And also reusability through being able to find that data back again. But very much Refget is part of the findable part of FAIR. Okay. This is about, it's roughly about halfway, so it's probably a good time just to pause for a second to make sure if there are any questions that have arisen or comments. If not, then I am quite happy to carry on. Sorry, I just realized as well that Ashley has said that they closed the application period as well, so I'm very sorry about trying to I'm very sorry about advertising that when that is actually closed. So if there's any questions, please pop your hand up. If not, we'll carry on. Okay, we'll carry on with going a bit deeper into Refget. Okay. So Refget version 1. Refget was conceived as a method of retrieving sequence by a checksum, as I said previously. We looked at a few other potential enhancements that we would like to Refget. One of these was to support subsequence retrieval, so being able to say, I don't want the whole chromosome of chromosome 1 because that's too big, but I just want the first 1,000 base pairs of chromosome 1. So we want to support subsequence retrieval. We also wanted to support a certain amount of metadata concerning a checksum. These are things such as length, known aliases, is the sequence circular, that kind of information. So very basic information about the sequence, but potentially important to know. We also felt that the API shouldn't be limited to genomic sequence, and we felt very strongly that Refget deployments could choose what they will distribute. Now, what we also knew is one of our major implementers of Refget was going to be the European Nucleotide Archive, which is based out of EMBL-EBI here in the UK. They actually developed a system which would work with the CRAM format to provide reference sequence based on an MD5 checksum. And so what this means is that the primary implementations of Refget would concentrate on genomic DNA, but we felt there was no reason to restrict ourselves to that scope. And you can see there's a link here at the bottom if you want to follow that offline to find more information about the CRAM reference registry implementation. So how did we go about designing it? Well, as I said before, we had the reference sequence on the left-hand side. We send it through this normalization process, which, as I said before, is A to Z only, uppercase and no white space. This normalization process was slightly fraught because we tried to bring together approximately 3 different normalization sequence algorithms, none of which completely agreed with each other. And the best halfway house we could come up with was support A to Z. What we really mean here, though, is that we're looking for support of IUPAC, DNA, and protein amino acid sequences with support for redundant sequences as well. So to be able to express, say, there's either an N in a sequence or a pyridamine or a purine or any of those ambiguity symbols. We then allow a implementation to go through 2 different checksums. The first of these is MD5, and that is there to maintain backwards compatibility with the CRAM reference registry implementation and with the CRAM format. So if an implementation wants to be compatible with CRAM, they must implement MD5 checksums. The second one was an algorithm called Trunk512. We generated a second one because we were concerned about the potential for collisions to appear in MD5. And so we wanted to future-proof ourselves slightly from that eventuality. Trunk512 is based on SHA-512, and then we take the first 24 bits of the resulting checksum and then convert that into a hex string. We do this for a number of reasons, one of which is to maintain compatibility with the Variant Modeling Committee's specification for reference sequence, which is an effort that I'm also part of. We also do it because if we detect a collision in trunk 512, we can just extend the number of bits that we're using in that checksum. And this also keeps the checksum string to more manageable volumes. Now, if we're talking about 100 or 200 sequences, these checksum string lengths are not so bad. And when we're also talking about reference genomic sequence, the size of the checksum string is normally negligible compared to the size of the actual DNA sequence that we're holding. When we talk about proteins and CDSs and cDNAs, actually, if you're going to hold the entire trunk, the SHA-512 in hex encoded strings, that's probably going to be longer than a lot of the proteins in the DNA and CDSs and cDNAs that we're actually going to have. So that's why we made this particular choice to do that truncation. What then happens is that these checksums are stored in a lookup database. That lookup database has a way to point towards the raw sequence. You come in with a URL that looks like this, so sequence / and then give it the checksum identifier. Here we're using the MD5, and then the lookup database finds that sequence and then fires it back across the wire as a plain text formatted string. So an example, this is actually two implementations. We have one which is called Refget, which is hosted off Heroku. This is a reference sequence, reference implementation that was written by myself and others here at EBI. We have a second implementation, which is the CRAM reference registry. Both of these implementations hold the Saccharomyces cerevisiae assembly R6411. And we have just said that this 6681 checksum is actually chromosome 1 from Saccharomyces cerevisiae. If you run the same query, both of these APIs return back the exact same sequence, which is here at the bottom. So you can ask either implementation for this sequence and you'll get the same response. We have a second endpoint available for metadata. The main difference between these two, if I can flick back, is you have sequence /sequence /checksum. All you need to do to get the metadata for that sequence is to add a /metadata onto the end of it. And here you can see that there is we allow a little bit of difference to appear between the two implementations. So Refget Heroku understands the alias I because it knows that it's chromosome 1. It could also have a concept of naming authority as well. So this could be set to INSDC. It could be set to UCSC, Ensemble. There are a number of known naming authorities that we've mentioned in our specification that we encourage implementers to use. But it's basically left there to be a fairly free text field. At the very middle, you can see that we have actually shared some metadata between the two implementations, which is the length of the sequence, which is exactly the same, and the MD5 checksum, which again is absolutely the same. At the very bottom, though, you can see that we do begin to differ again, and we've both decided that we will not provide. So Refget provides the trunk 512 checksum for this sequence, whereas the ENA implementation does not provide that. So again, it's up to implementations to choose what they want to support. Now, one of the things you might say from that is, this is really difficult to know what a service actually implements. To solve that, we have another endpoint called service info. Service info will provide information such as the algorithms that a service supports. So here, this is from the Refget Heroku version. And you can see here that it supports MD5, trunk 512, and another naming algorithm called VMC Digest. You can also see certain capabilities of the server being expressed here. So Circular Supporter says that my implementation that's available, you can make circular sequence calls. So you can make calls where your starting point is at a greater position than your ending point. And so if you're on a circular sequence like a bacteria, you would go through the origin of replication and retrieve back all of that sequence. There are other potential limitations as well. So there may be a maximum to the amount of subsequence you can request from the API, and so we would want to transmit that. And also there's a reason to send over the wire that there is what versions of the API this implementation actually supports. So this means that you can make more intelligent clients. You can know what you can provide to a client and how, sorry, as a client, you can know what kind of requests you can make to a server and what you should quite reasonably expect back from that server. More generally, we are looking towards expanding this out across the global alliance APIs as well, and we are starting a piece of work on doing that. If anyone is interested in that as well, then please also get in touch with me and also with Rishi, because this is something that we would like to normalize across the entirety of our APIs, or at least normalize the mechanism by which you get this. In terms of deployments and interoperability, so we have the specification, as I said before, that's currently located under the HDS specs API under refget.html. We have two actual deployments, which is the Refget Heroku app, and we have the CRAN reference registry. The CRAN reference registry is the only production quality release of Refget that we have at the moment. The reference Perl implementation is there as a proof of concept for the specification. We are currently also working with AWS to develop a serverless version of the Refget API, which will mirror a huge amount of the CRAN reference registry data, and will also provide not only MD5 checksums, but also trunk 512 checksums as well. So that will be a part of Amazon's public dataset project and will be available in multiple availability zones. So you will be able to go to your local Amazon instance and start retrieving data from there. We've also had interest from Google and from Microsoft Azure to actually deploy similar versions into their own cloud as well. So hopefully you'll start, if you're running cloud workflows, you will be able to just use cloud and your local deployment of Refget to get your reference sequence. You won't have to come back to EBI for that information, which is good, because if everyone around the world tried to retrieve sequence from us from EBI, we would not be able to provide all that information at the same time. In terms of interoperability, now, because we have so many different versions of this we spent a large amount of time with a Google Summer of Code student to write a what could be thought as torture tests or edge cases, where we know that APIs must behave in order to provide compatibility between implementations. So you can see there is a set of compliance documents available from Read the Docs. And also we have a nightly compliance report, which at the moment only runs against 2 versions of the API. You can see a screenshot from here, but basically you can see that both the Refget Heroku app and the EBI implementation passes all test cases. We have the service info for metadata for sequences and specifying sequence ranges. And also our compliance report as well is capable of using the service info endpoint to know if an implementation should or shouldn't pass a test. A good example is here. This is a screenshot from the EBI implementation from the CRAN reference registry. CRAN reference registry does not support circular sequence queries. So we do not check to see that it supports circular sequences retrieval, because we know it would fail. So we just pass that and warn and say, this is okay. The server implementation doesn't fail, it just doesn't implement that particular capability. Okay, if you want to use it today, as I said before, you should use the CRAN reference registry implementation. It is the most comprehensive deployment of reference sequences. It's based on ENA, and so it has most of the sequences submitted to INSDC available through it. There will be some bits of INSDC, there will be some sequences that you won't be able to retrieve out of that, but for all intents and purposes, you pretty much have access to all of INSDC. And INSDC, for those who are not aware of the acronym here, INSDC is the 3-way collaboration between the European Nucleotide Archive, EBI, GenBank, based at NCBI, and the Data Bank of Japan as well. Okay, so a little bit on Refget's future. So I said before that we are currently working with AWS to deploy a cloud infrastructure for this, for Refget. This is a piece of work that is ongoing, and we encourage people to come and join into the work stream as well to help us work through this. The basic idea behind this is that we want to look towards using serverless technology. So the idea here is that we would have a Lambda process which would represent the Refget implementation, which is what's available from this GitHub repository at the top left. We would store our metadata in a DynamoDB and key that by the various checksums. And then we would actually store the underlying reference sequence in Amazon S3 as a public data set. And we store that as a single line of sequence without any line breaks. So when a user comes in, we could either serve sequence back from the Lambda process directly, but there's a maximum payload of this of 6 meg. That's just a limitation of Lambda that exists. So we would, this is where the subsequence limit service info endpoint comes into play. You then know I can't make a request more than 6 megabases, so I won't attempt to do that. A request comes in, the Lambda process finds the metadata. That will tell it where that reference sequence is held in S3, and then we do a redirection of the client back to S3 to say, please go over here for your reference sequence. This is all done as part of Amazon's public dataset service, and we think that this is actually going to be a really good sustainable way of providing reference sequence in the cloud that basically plays to the cloud's greatest strengths. Now, one of the problems once we deploy to the cloud is how do we actually know which implementation of Refget to redirect someone to? So there is a question here about federating each of these different deployments. Within the different EMBL-EBI, AWS, Google Compute, and Azure proposed deployments of this, each of these different engines will effectively be equivalent to each other. Sorry, not engines, deployments will be equivalent to each other. So because of that, we are considering part of GA4GH's discovery work stream is to come up with a discovery network that would allow us to do redirects, say, based upon geolocation. So making sure you get forwarded to your closest implementation of Refget. It may also be that we have implementations that maybe don't have all sequences available, or maybe they're missing on certain pieces of sequence. And so another level of this could be to say, well, I've gone to AWS, that doesn't have this particular checksum, but then the network is capable of actually routing through that to find another location that could satisfy the request. We are still throwing these ideas up in the air at the moment. The first example definitely is the way we want to go first. The second example about intelligent routing based on existence or non-existence of an identifier is something else to look into. If you want to be involved in Refget for this work, this is happening as part of the discovery work stream. Another piece of work we're looking at is the idea of sequence collections. And I want to take your mind back to one of the very first slides I showed in this webinar. And that was the idea of how reference sequences are fundamental to genomic analysis. Now, obviously, none of you who have done genomic analysis have a single chromosome and align against a single chromosome. You align against a genome. And if you're doing, say you're using Callisto, you use a transcriptome. That means that reference sequences do not exist in isolation. They're not an island. They are part of a coherent analysis unit. We know that this is a big thing that downstream users are going to need. It's not good enough just to have provenance of the reference sequence that something was aligned to. It also matters what was the analysis set, what was the actual reference set of sequences that were aligned to. As a very concrete example of this, one might look towards downloading the GA4GH38 assembly from the 1000 Genomes project. You may want to supplement that with the HLA regions, with the GRC patches. You may also want to supplement that with the decoy sequence as well. And so it really matters what you aligned against, because you may look at input data coming into your own analysis systems and decide that the checksum, as the sequence collection that was used, was insufficient for your own levels of quality that you have in your own pipeline. So this is something that we want to do. We want to capture these sequence collections. We want to capture their metadata. So what are they called? What they're commonly known as. It's a work in progress. We encourage participation. But there's something of how this might work. One could consider that we have, say, 4 examples of sequences here. It would be possible to sort these different checksums, concatenate them together into a single string, and then pass them back through the same underlying algorithm that was used to create those checksums. So this was the trunk 512 algorithm. These sequences are already sorted lexicographically. They've already had the trunk 512 algorithm applied, and out pops this new identifier, which uniquely identifies this collection of 4 sequences. This is a possible way that it could work, but there are obviously some big questions to answer here. So things such as, how do you know if there's partial overlap between 2 different sequence collections? How would we scale this sensibly towards, say, an entire transcriptome, where there would be, for human, perhaps in excess of 100,000 of these checksums existing as well? I believe this is all solvable, but it is an important thing to consider. Okay, so that's the end of the webinar, at least for the presentation section. In summary, Refget provides access to reference sequences, subsequences, and reference sequence metadata through a standard REST API. Refget is a foundational API Global Alliance. It's one of the raw ingredients for genomic analysis, and all of our future work that we are doing as part of Refget will provide a robust mechanism to define reference analysis datasets and allow a network of APIs to operate within the context of Global Alliance and within users of that API. And just to briefly send out some acknowledgements for this, this is not just the work of myself, this is the work of a huge number of people with support from a huge number of different institutes as well, without which the specification would not be in the state it's in today, and without which we wouldn't have a Refget implementation as well. With that, I'd like to say thank you to everyone for your patience, for your attention, and I'd be quite happy to open this up to any more questions. I believe we've got a hand up from David.
David, I'm having trouble letting you talk, so if you could type the question to the chat, that would be great.
Hello? Hello.
Hey. I can see as well we have some representatives from the Refget group as well here as well, and future contributors as well in Lisa and in Thomas as well here. And so thank you guys as well for joining in. Ah, fantastic. That is a really good question. Okay, so hopefully everyone's got the chat window up. Thank you, David. That's a brilliant question. Is there any plan to support annotation to those references in the Refget API? I would say within the context of Refget itself, there is no plan to support annotations, purely because we feel that we need to maintain a tight specification and a tight implementation behind this. Thomas is just, attendees can't see questions. Okay. Hopefully everyone did hear the question though, is there any plan to support annotation to those references in the Refget API? So Refget itself will not support that, but what we do support is other third parties coming in and using Refget and its coordinate system, the ideas of this checksum system to provide annotation based on this. As I said before, my day job is more involved with the Ensemble project. Ensemble is one of the major annotators of the human genome. We provide gene annotation, we provide variation annotation, regulation annotation. Refget is very much part of our plans, and I very much see being able to come in with Refget checksums and retrieve annotations based on that Refget checksum identifier to retrieve annotations. And this is really as part of our future development plans here at Ensemble. How much this will be picked up by other people, I don't know. Okay, second question. Hopefully that answers your question, David. Okay, second question is, could standards and APIs like Refget be developed for other kinds of genomic data? For example, SNPs, indels like dbSNP, or expression data like from GTEx. Is this on the roadmap? There are definitely other efforts to develop similar ideas behind similar concepts of checksumming as we've seen as part of Refget. The VMC specification very much, which is Variant Modeling Committee, part of Genomic Knowledge Standards, has at its core the idea that you can create a checksum digest based on the observed event. So say a SNP or an indel, and from that you could generate a unique checksum which could then be used to actually verify the integrity of that variation and also potentially exchange variation between two different resources. In terms of expression data from GTEx, I am not aware of that. What I do know though is that we have had a bit of interest from Mike Love, who is actually one of the maintainers of Callisto, if I remember correctly, and he is developing a number of equivalent checksum ideas to define reference annotation input into Callisto. So I can't say that there is an idea of using similar standards to APIs like Refget in expression data, but there is definitely an idea that Refget could form part of that tool chain. Hopefully that gives you an answer. I think you've dropped off. Oh no, you're still in. Okay. Hopefully that answers your question. Okay, I can see everyone is dropping off now bit by bit. Oh no, people are coming back in. Are there any other questions from the attendees? I think Thomas is trying to talk. Go for it if you can, Thomas. And thank you for the kind words, David.
I don't know if you can hear me.
Yeah, I can hear you at least. Hopefully everyone else can.
I wasn't actually trying to talk. I was just messing around with the settings here. But I suppose as the opportunity arises, you know, one thing we're thinking about is that, you know, Refget is one way of looking up by checksum. There is something that we think about the European Variation Archive is that we need a kind of accession MD5, accession checksum, commonly used identifier sequence resolution service, or something along the lines. Like checksum is one way, you know, then there's the multiple different accession codes for the same sequence, and that's another way that people look up sequence or need to be able to find sequence. So something that we're thinking about is, you know, having some kind of service as well as Refget, which is checksum lookup, but also having a kind of a mapping of all the different accessions that the sequence is known for that given sequence, along with the commonly used identifiers that are also used for sequences, and having a kind of sequence resolution service that has those multiple different methods of lookup.
That's a really good point, that is, and that goes back to one of the earlier slides as well, with the naming problem and how does, and it's nearly a catch-22 situation actually with Refget. How do you get hold of the checksum? You need to have the sequence, and how do you get hold of the sequence? Well, you need the checksum. It's nearly it's like a Gordian knot really that needs to be.
I mean, ideal world is that the header of any type of file, a BAM file, a VCF file, any type of file that's relative to a sequence, a GFF, that it's ideal world is that it's sort of the checksum is mandatory to be included, but that's not the reality, I think. So, yeah.
Yeah, so I think there's engagement with tooling required here to make sure that tooling does provide these checksums, but also we need to help along the way as well to actually shortcut that process.
So actually, that's kind of a conversation we should try and mediate. Actually, the main sort of tool chains that we know that we have contacts with, like the GATK and the SAMtools people, that if we were able to get them to by default output those checksums, then that would sort of certainly be more of an enabler for Refget use in the future.
Yeah,
somewhat solve the chicken and egg problem.
Especially if we could also have support for collection, for the sequence collection, whatever format they take as well within this.
Yes, sequence collections is the other thing. Yes, because one thing we started to use we were starting to look at using was GCA numbers for submissions to the EBA, but of course GCA numbers are not, they don't include the mitochondria, because that's often submitted separately, and then people make up their FASTA, separately including the mitochondria, and then So when you talk about collections, I guess you're thinking of ordered checksums.
Yeah, that's currently the thinking, but obviously this is early days, and obviously people on here as well are very welcome to join in the conversation and basically drive what this is going to mean. Okay, I can see that we're 5 minutes to the hour. If there's no other questions, I'm quite happy to stop it to bring the webinar to an end. Thank you very much for your time. Thank you very much for your questions as well. And thank you basically for turning up, and have a nice day or a nice evening, depending on where you are in the world.
Thanks, Andy. All right, with that, we'll end the meeting. Thank you all for joining.